So I'm George Koob. I'm the director of the National Institute on Alcohol Abuse and Alcoholism, and I'm a senior investigator at the National Institute on Drug Abuse.
Well, thank you for talking with us today, Dr.
My
Koob.
pleasure.
So I wanted to start by asking a bit about your background. I've been asking each person how you got to where you are, and I wanted to say you've traveled extensively. You lived in France, and did you see that your upbringing affected your career?
Yeah, sure. My father was ultimately a colonel in the Army. He was part of the invasion of Okinawa in 1945 and stayed on Okinawa, where he won a battlefield commission. And my mother earned a Ph.D. from NYU in 1946 in English. And so she came out, and I was born on Okinawa, where my mom founded the dependent school system that's still there. And then we returned to the States, and I basically lived somewhere. Every 2 to 3 years we moved for most of my adolescence. And we settled in Pennsylvania, where my dad was comptroller of the Letterkenny Army Base. And so that's where I ended up going to high school and college. And then ultimately, ended up at Hopkins, Walter Reed Army Institute of Research, postdoctoral stint at the University of Cambridge with Susan and Leslie Iverson, and then moved to the West Coast to join Floyd Bloom's lab at the Salk Institute, and never looked back. So that's my life story in one sentence.
And there's a lot in there. There's a lot in that brief description. In your education through high school, college, are there particular courses that you enjoyed and found influential for where you are now?
Yeah, because in high school I had a teacher who taught me biology, but also Latin, and she was very charismatic. And so that was the time of, Watson and Crick had really just published, not too much earlier, the double helix. And so it was an explosion of interest in biology. And I was always trying to juggle whether I wanted to be in the classics, and considering, and I actually enrolled in Penn State in classics, but at the last moment before I went on campus, I switched to biology. And ironically, my brother Steve ended up being a classics major, fluent in Greek, and became a famous archaeologist conservator of glass. So he's still around and still famous.
I think in a little bit we'll come back to the conversation about your brother. And how did you end up pursuing behavioral physiology in particular?
I wanted to be an ethologist. So I wanted to, basically watch animals in their natural environment. I had read all the early ethology books, and was just totally intrigued with it. And when I was at Penn State, I went to the psychology department to ask about a professor whose name I don't remember anymore. And I don't think I couldn't track him down when I did did a biography, an autobiography for Society for Neuroscience. So all of what I'm telling you is basically published in that autobiography. But that was a big transition point, because he was this professor at a joint appointment with the University of Georgia and worked on in primates in the Caribbean, and he was away that year. And so the professor of psychology sent me to Howard Hoffman. And Howard Hoffman occupied an old building on campus that was originally a part of a farmhouse, I'm assuming. And he was training ducklings to imprint to a milk bottle on a train track. And so he had these ducklings living there, and when at the right imprinting moment, in a Conrad Lorenz kind of way, he brought these ducklings upstairs and put them in this enclosure where a white milk bottle went back and forth on a train track, and that became Mom. And I was like absolutely fascinated. And so I asked him whether I could do an independent study working with him, and he said yes. Stuck me in a corner with Skinner's Schedules of Reinforcement, and said, "Read this." And so began my career in behavioral pharmacology, even though we weren't doing any pharmacology in those days, or physiology, behavioral physiology, I guess you would call it. And so, from that point, I applied to graduate school in ethology at heard of 3 or 4 institutions, and I got in all of them. But I got an interview at Hopkins, and they had a pathobiology program in ethology. They had an ethology program in the pathobiology department at Hopkins and at School of Public Health. So I went down and I did my interview with them, and they explained to me that they'd love to have me join the program, but they had no more room on their training grant. They had a T32, or it could have been an NSF. I'm not entirely sure which one, funding agency supported it. This was early 1969, and there's another complication. The Vietnam War was ongoing. My dad had made it very clear that I was to join ROTC at Penn State, so I did. And so I had found out there was a delay of active duty program to pursue a Ph.D. with the Army. So that was the plan. So I graduated early from Penn State in March, I think it was, April. Went down to Hopkins. And as I said, the pathobiology department didn't have room on their training grant, but they said, upstairs in environmental medicine, which we have a good collaboration with, they have room on their training grant. So I went up and I met, I think his name was Richard Riley, who was the chair of environmental medicine and MD, one of these classic Hopkins professors with the skull writing across his white lab coat. And he gave me an interview, and he said, Koob, there's a few C's here in your transcript. I don't know what we're going to do about the C in map reading. And that was ROTC. I couldn't really get into some of the ROTC stuff, having lived in the Army my whole life as an Army brat. And so I said, yeah, well, I really couldn't get into it. And he said, that's okay. He said, I'm against the war. You're in. So I was trained on a heart-lung, NIH heart-lung training grant in physiology, not neuroscience. Neuroscience didn't exist at that time. And that was where I did my PhD. So that's how it all evolved, education-wise. And I had a wonderful time at Hopkins. I met Candace Pert on the bus on the way to Homewood campus, the other campus, and she wanted to organize all the students who were working in neuroscience before there was neuroscience. I went to the first Society for Neuroscience meeting in Washington. I saw Huda Akil give a talk that was so prescient because it basically predicted that the conclusion of her talk was there had to be some morphine-like substance in the brain. This was long before Saul Snyder had identified opiate receptors in the brain, or Kosterlitz, and Hughes had identified endogenous opioid peptides. And so it was a very exciting time. And being at Hopkins was like a dream, because, you know, there was people like Phil Bard, who read my first paper, Phil Bard of Cannon and Bard, Kurt Richter, who was identified as the father of physiological psychology, you know, Vernon Mountcastle, um, and other people who, you know, at the time were probably in their 80s and 90s. So I got to see some of the towering figures of what became neuroscience.
As I listen to your description, it really drives home a point to me about there being key people, key moments in our careers and in our lives that have an enormous influence. Would you mind, maybe a little bit of an aside? You mentioned your brother before, but tell the story about the creation of the word hyperkatifia.
Yeah, so I've always been, well, I'm going to predicate that with a bit of a long story. So I wrote about these in the autobiography. I call them eureka moments. So there were periods in your career where something changed dramatically, and it was usually associated with finding a result completely the opposite of what you expected. And so it wasn't actually working on alcohol, I was working on opiates. But I had been working on alcohol and opiates and psychostimulants and their rewarding effects for probably about 15, 20 years. So I contributed, I wasn't the only one. There were herds of laboratories working on the rewarding effects of drugs. But I was curious as to, I'd read the work on opponent process by Richard Solomon, and I was curious about the after effects. And so with Tammy Wall, who's now, I think, vice chair at psychiatry at UCSD, she was an independent study with me, an undergraduate. We decided to identify where in the brain opiate withdrawal took place. And the literature said the hypothalamus, the periaqueductal gray, the, you know, medial thalamus were all these sites. To make a kind of short story long story short, I used an operant technique to measure withdrawal, which is suppression of lever pressing during precipitated withdrawal. And I decided to precipitate withdrawal by micro-injecting methyl naloxone into the brain. So we micro-injected it into those three areas. And, you know, low doses had no effect. We had to move all the way up to like something like 4 micrograms to get an effect. And I had thrown in the nucleus accumbens as a control site. And when we micro-injected in the nucleus accumbens, the rats completely suppressed their responding. They were completely, quote unquote, dysphoric-like. And we had to go down to 4 nanograms to even get a threshold effect. And then all the light bulbs went off in my head, because I realized that it was the same system that makes you feel good that makes you feel bad. And so that got me really into opponent process. And I spent the rest of my career trying to understand, you know, why drugs make you feel bad. And so I got into came to realize that it wasn't anything to do with physical signs of withdrawal. I hate that term, because the brain's not physical, right? It had to do with malaise, dysphoria, you know, irritability, sleep disturbances, pain, emotional pain. But I got tired of writing, you know, hypersensitivity to a negative emotional state associated with alcohol withdrawal or opiate withdrawal. And so with 2 anesthesiologists in 2010, we were getting an inkling in 2010 that there was something going wrong in the opiate world in pain control. And so we wrote a paper. It was Howard Gutstein and uh, Joe Sherman. Howard Goodstein at the time was at MD Anderson. He's an MD anesthesiologist. Joe Sherman is a famous anesthesiologist in the San Diego area with private practice. He treats the rich and famous and anybody. I mean, he's a wonderful, real, anesthesiologist. And I got tired of writing hypersensitivity to a negative emotional state, so I called up my brother Steve, who speaks Greek fluently, and I said, I need a word for dejection, sadness, dysphoria in Greek, and he gave me katifia. I just slapped hyper on because I knew the word hyperalgesia, and I figured I'll just make a parallel word for hyperkatifia, which would be, instead of hypersensitivity to physical pain, it would be a hypersensitivity to emotional pain. And we sent the paper in, and the review came back. One of the reviewers said, well, hyperkatifia doesn't exactly roll off the tongue. So we had a choice, and one thing we raised amongst ourselves was that we just dropped the word because we wanted the paper accepted. But I wanted the word in the literature. So I wrote back, which I don't normally do. I don't usually, try to confront the editor in any way, but I pointed out that hyperalgesia doesn't exactly roll off the tongue either. And they took the paper and it was published and completely ignored for 10 years. Well, not quite 10 years. So it was probably around 2016. I was giving a talk at NIH, and I actually, it was in one of the big auditoriums, but I didn't realize even back then that it was being webcast across NIH. So in some sense, NIH was a little bit ahead of its time. This was pre-pandemic. Zoom hadn't come into the picture, but they could do web casting. And so I introduced hyperkatifia there, and then it started to take off. And I remember one of my fellow IC directors really liked it. And the director of NCATS, and I'm always blocking on his last name, but it was Chris. I'm sorry, we couldn't remember his name last night, but I'll think of it. In any event, that's how hyperkatifia came about. And then I published a paper fairly recently. It was 2019, during the beginning of the pandemic, in biological psychiatry, where I put it in the title, and then it really took off. So that paper's been cited almost 400 times.
But personally, I think hyperkatifia is easier to say than hyperalgesia, at least in English.
Well, it's a word that's quite warmly embraced by clinicians because they see it in their patients. I worked at a treatment facility before I came to NIH. I would go down once a week and talk with the patients about what happened to their brain, and they would be totally unresponsive the first week they were in the it's a Betty Ford like outfit. And they basically did everything from motivational interviewing and cognitive behavioral therapy to all the medications to biofeedback. But I watched the change over each week in the patients, and by the fourth week there, most of them had relaxed, opened up, and were full of questions. But the first week they were in hyperkatifia. And so, it really struck home. But I still find that basic researchers have a little bit of problem because, the field is still dominated by reward. But what I really think drives addiction, moderate to severe addiction or moderate to severe alcohol use disorder, is not reward, it's relief. And as I get older, I can become more and more convinced that it's a major driving force. It's not the only one, but it was. I think I can safely say that it was completely ignored until about 20 years ago.
I think that the word itself is actually very important because language affects how we see the world. And when there's a word for it, I think that over time that will enhance people's perception of the concepts you're describing. So would you please talk about the vapor chamber model? It's something that you and your group have used extensively.
Yeah, so when we were at Salk Institute, Floyd won an alcohol center grant. And so we had one of the original alcohol centers. And I might add that it's still there. So after Floyd directed the center, I took it over when he joined Science, became the editor of Science, and then Larry Parsons took it over, and then subsequently, when Larry died prematurely, Barbara Mason took it over. So it's still part of the NIAAA grant portfolio. And I believe there are two other centers that are equally as, well, we're part of the original site, the one at the University of Connecticut Medical Center, and that Victor Hesselbrock has been directing. I think he's stepping down now, but and the other one is Indiana University, still has an alcohol center. But you know, when we started, we wanted to make the animals dependent, and so we tried. Tony Deutsch over at UCSD, who became a very dear friend and introduced me to my hobby of growing fruit trees, had developed a model where he put in intergastric cannulas and gave the animals alcohol through their gastric cannulas, and made them dependent, and then they would actually self-administer alcohol that way, ultimately. And so we tried that. And Joe Rogers was a postdoc, and I was a staff scientist, and Joe and Sandy Weiner tried the intergastric method, and it turned out to be, well, in those days, you know, we didn't have the veterinary support that we have today in our work, and so we had a lot of problems with infection with these catheters in the stomach, and it just got to be unwieldy and not very productive. And then Dora Goldstein had been giving vapor to animals, to mice, but using pyrazole to block, you know, alcohol dehydrogenase. And we wanted a cleaner method, as I remember. And Joe came up with the idea of exposing rats to vapor. And so they published a paper in 1979 that came directly out of the Alcohol Research Center that Floyd had started. And the paper is Joe Rogers, Sandy Weiner, and Floyd Bloom. And Joe went on to a career as a senior scientist at Stanford Research Institute, SRI. And we kept the chambers as part of our center for years, still are. And so we utilized them. And it was a way to efficiently make rats dependent on alcohol. And then we devised a technique to test them during withdrawal, either 6 hours or 12 hours into withdrawal. And then the animals, these are all male rats at the time, would drink a huge amount of alcohol. I mean, in 30 minutes, they would drink the equivalent of about 4 or 6 glasses of wine. And, you know, we published on it extensively. And then it started to catch on a little bit. But we got a lot of grief about the vapor model because people said, well, it doesn't have any face validity. But, you know, face validity has its limitations when you're talking about rodents. And I told Sponigal that once when he was complaining about the face validity. Ronye Sponigal asked me a question at a big congress once. There were a couple hundred people in the room. And he said, well, you know, what's the face validity of, you know, it's not natural for rodents to be exposed to ethanol vapor. And he was going on and on. And I said, Ronye, what you want is a rat standing on a street corner holding a bottle of bourbon paper bag smoking a cigarette, and you're not going to get that. And so we went on, and then Maury Cole was hired to run the chamber system for the Alcohol Center, which he did for many, many years, and certainly under my tenure, and did an outstanding job. But then he, with Catherine Riviere, branched off and founded a private company to market the alcohol vapor chambers. And he has now, I think, sold alcohol vapor chambers to practically every country in the world and every state in the union. And he's still doing it. And, you know, he's been very successful. He has a booth at RSA every year. And he's been extraordinarily successful. He also works on vaporizing, you know, other substances of misuse. And so, so that's how they evolved. And then they've been really validated as a predictable model for alcohol dependence. And even, you know, I mean, if you want face validity, Olivier George actually and his team have trained animals with Maury. I think Maury did it with them. They've trained animals to self-administer alcohol vapor if they're dependent. So, you know, I mean, and there was a period when human beings were actually vaporizing alcohol in bars. It's been banned in most states because you know, the amount of alcohol you get is not well controlled in that context. But so that's the story of the alcohol vapor chambers, and I think they're probably pretty widely accepted now. Every once in a while, some reviewer will give somebody a hard time, but I think, you know, it's like the conflict test that was developed many, many years ago. I mean, you know, that was, you know, where the animal got an air puff or a mild shock to their feet when they got a reward, highly predictive of anxiolytic drugs like benzodiazepines, but it's certainly not got much face validity. How many human beings are sitting in a room, you know, getting air puffs to their face and, you know, needing a benzodiazepine?
Yep. Those are good points. When when I hear people say, I have the only way to do something, or you can never do it that way, I get suspicious. I think it depends on what you're trying to find out. The vapor chambers have certainly been very useful. Do you talked about eureka moments? Do you have any other eureka moments that you would like to describe?
Yeah. So you know, one was when Wiley Vale and Catherine Riviere and Jean Riviere and Jacob Spies sequenced CRF, which would have been, I think it was 1981. I was at the Salk Institute. And so they asked me to test CRF. And it was a 41 amino acid polypeptide, so you couldn't give it systemically. We had to put it into the brain. And Marvin Brown had already looked at physiological responses like high blood pressure and things of that nature by injecting it into the brain, but no one had observed behavioral responses. So I remember the day so clearly because we had a bank of 16 photocell cages that we used for locomotor activity, incredibly simple, with two beams across the long axis. And you just measured how many times they broke the beam. And we had some small male rats again, and we injected them with CRF in a dose-dependent fashion. And I called up Wiley because it was over in my lab and he was in the annex. And I said, you got to come over here. We injected the rats with CRF and they're levitating. And so he came over and we watched what happened. And I'd never seen this before, but at low doses, the animals were climbing the cage and very activated. But at high doses, they were showing forepaw tremor. Well, in the end, we then went on when Michel Lemoyle came over for the summer. We went on to inject them and test the hypothesis that CRF was an arousal stress neuropeptide in the brain, as well as what we knew about it driving the HPA axis. So this was independent of the HPA axis. And that was our hypothesis. We actually wrote it in the grant proposal, that it's a neurotropic effect in the brain to potentiate, to mediate behavioral responses to stress. That was the hypothesis. And so it all folds into a kind of eureka moment, because we then tested the animals in an open field and found that the same doses that caused the behavioral activation in a familiar environment produced a complete opposite effect, a stress-like response in a novel environment. And we published that paper in Nature, and the rest is kind of history. But there was a second eureka moment built into it, which is when we first tested one of the peptide antagonists. And this is really critically important because everything we had shown was pharmacological effects of dumping a peptide into the ventricles of a brain of a rat. But when we injected a CRF antagonist, it was alpha helical 9 to 41, into the brain of rats, and we blocked a natural stress response, then we knew that the hypothesis was actually correct, because we were blocking endogenous CRF. We weren't blocking anything we'd stuck into the brain. This was like giving naltrexone or naloxone and blocking endogenous opioid peptides. And it's so funny, to see how things evolve, because even now, I remember when naloxone was considered completely and utterly inert, because the behavioral pharmacologists were using schedules of high responding, like VI schedules, variable interval schedules. They gave rodents naloxone, had no effect. Nothing happened. It wasn't until the opioid peptides were discovered in the brain, opioid receptors first, and then opioid peptides discovered. And then people started seeing effects of naloxone on endogenous behavior. And it's an old principle that Thomas Hochfeld, who was one of the pioneers of identifying neuropeptides, argued is neuropeptides are transmitters that don't work in the quiescent state. They work in an activated state. In other words, they have a role in promoting arousal or promoting hunger, or where the organism is engaged in some major activity. But if you're just hanging out, CRF is not activated. But if you step across the street in Georgetown, Washington, D.C., and almost get hit by a car, because how the driving is in Washington, and then you step back, your whole physiology has changed. That's CRF and other neurotransmitters that are activated. And then, of course, later, people discovered that CRF was in the extended amygdala, marked the extended amygdala, huge amounts of CRF neurons and projections associated with the extended amygdala. So it all came together. So that was another kind of eureka moment. I'm trying to think of other ones that that really, there let's go on to something else. I'll try and think. I mean, I have about 5 that I think of, but I mean, those are 2.
But we can come back to it. Yeah.
Yeah. So I mean, one was when we discovered opponent process. The other was the CRF system. I guess it was a third part of that is identifying CRF and other stress-related neurotransmitters being capable, when you block them, that you actually block dependence-induced drinking. I think that was a big, dramatic change, and selectively in some cases. In other words, these antagonists like kappa antagonists and CRF antagonists and hypercretin antagonists and norepinephrine antagonists, they don't have much of an effect in a non-dependent animal, or and in a dependent animal, you're actually reversing this relief drinking. And that was a big breakthrough, I think. It's not completely black and white, because if you crank the dose up high enough, you'll see effects in non-dependent animals, but there's a big difference. So that was another one.
Oh. And a sharp turn in direction. What you've described is a lot of basic research in rats. And something that struck me immediately when you became director of NIAAA was your attention to getting the word out to the public, really getting the information to people who need to know it. Um, is that something that how long had you been thinking about the need to pursue that? How did you come to do that so quickly?
Well, I think it probably preceded coming to NIH, because I started a collaboration with Barbara Mason when she was at the University of Miami, and I remember it fondly, because I remember she wrote to Amanda Roberts and said, "Can't believe I'm going to be collaborating with someone who works on those icky rats." And Amanda Roberts wrote back and said, "I can't believe we're going to be working with someone who works on those icky humans." I mean, we're all friends, so it was all in jest. But I think you come to a stage in your life where you want to see what you've done translated to help people. And so that bug had already been in me. And, we even had teamed up with a number of people to see if we could actually start a company to look at medications for the treatment of alcohol use disorder. Unfortunately, the gentleman we started to work with, who was wonderful, got a brain cancer and died. And, you have to have a special person to work with if you're going to do this kind of stuff. And so it never worked out for us, for multiple reasons. And then about at that time, when we would have been maybe trying to find somebody else, the call came from NIH, and so I got to NIH, and I I started, basically the way I decided to learn about what the institute was doing was to have people who were giving presentations for NIAAA give me their slides, show me their presentation. I mean, not come in my office and give me the talk, just send me the slides and let me start looking at what we're presenting and what, and it really, and then at the same time, we were challenged by the powers that be to do the collaboration on research on addictions at NIH with NIDA. So, we were told we were going to work with NIDA and do things together. And, I had already been working with Nora Volkoff for years on preclinical things, but it came to understand some of the things that we were doing in that regard. The NCANDA study had taken off on cognitive and neural development in humans, a longitudinal study. It was the precursor of the adolescent brain cognitive development study. All this stuff was starting to churn in my head, and I realized how much we knew and how little the public knew about what we knew. And I realized that, we had an enormous alcohol problem in this country. I probably hadn't realized how enormous it was. And it's still, and it remains. I mean, it's endemic, you've heard me talk about it. And so all of this came together, and I realized that, people had no clue about alcohol, and the medical community, and I went to a meeting of all the college, about college teaching addiction in medical schools with NIDA. And so I went, and NIDA representatives went, and they were all sitting around a room, on this big table, all these deans and whatever they were called at their medical school. And they said, of course we teach addiction in medical school. And then they came to the lady who was the president of the Medical School Association for all medical schools. And she said, no, you don't. That we actually have to form our own mini seminars at lunchtime out on the quad and try and get some instruction on what to do about addiction. And when it came to me to speak, I said, how many of you in this room can name the 3 medications approved by the FDA for the treatment of alcohol use disorder? And they all, started scuttling under the table with their, at that time, it wasn't an iPhone, it was probably one of those other things, And then I realized, we don't teach addiction in medical schools. That means primary care docs don't know anything about alcohol. And it just all came together. And that's when I started everything, and also, my predecessor, Ken Warren, who was the acting director, before I became director, had started with the team, and maybe it even started before Ken because it took them some time, but they had produced College Aim, which was this menu of prevention for colleges and universities. And when I came on board, I was asked to help sell it with the college presidents, which I did. And it turned out, I found out it was excellent. And then I realized if we could do it with that, we could do it with many other things. And so I started, it was my idea to, if I may be so narcissistic, it was my idea to produce the NIAAA Treatment Navigator. The healthcare professional core resource was also my idea. And we're not done. We're working on what I hope will be a 30-minute core curriculum presentation for medical schools. It won't be done for some time because we only have the first draft right now, but I'm aiming for like 30 to 40 minutes total time. And we still have some other things in the works. I'm trying to get some kind of core prevention resource out there. We have a Native American prevention called Native Air that's going to be launched in the pretty near future. And so, the whole effort has been directed at getting the evidence-based information out to the public. And in the same token, that allows us to also refine our strategic plan, which I presented yesterday or the day before here, and helps us focus our goals on what's needed to be done. And so out of that evolves, women and alcohol, aging and alcohol, fetal alcohol spectrum disorder, all of these areas where we just need an enormous amount of work. New treatments for alcohol use disorder, behavioral or medications, and health services. And one of our new themes, I'll probably talk about it next year at RSA, is alcohol is the 5th vital sign. We want to get screening, brief intervention, referral to treatment in every doctor's office, every health clinic in the country. And we do have screening, but we don't have any referral, any intervention or referral to treatment. And motivational, you can do a modicum of motivational interviewing in about 2 to 5 minutes. And it doesn't have to be the primary care doc. It could be the person who intakes you. It could be a nurse practitioner. It could be, someone with an iPad or a video. There's so much that could be done that we're still not doing, in my view. So, that's where it came from. It was a evolution of and convergence, and I guess you could call it a eureka moment. Another one of them, but more my NIAAA one, when I just realized that, Americans and the healthcare profession just don't know much about alcohol.
Yeah. Thank you for that. It's someone who My wife says I'm a doctor, but not the kind that helps people.
Right.
So it's exciting to see the effort to get this out, the use. And what would you like to see, and what do you expect to see in the future of alcohol research?
I hope to accomplish some of the things that we've been talking about. So I would like people to understand, particularly young people, and I think that's where we're seeing changes. That there is in general a cultural shift going on. Whether we'll stick with it, I don't know. But we had a 20-year decline in underage drinking. These are the kids that are embracing, dry January and sober curious and mocktails. Mocktails are now, you can find in almost every restaurant. And even the Wall Street Journal had a front page article about a rather tasty non-alcohol containing beer that seems to be the rage and taking off. So I think there's a shift going on. I'd like to see alcohol understood in the context of what it's used for as a social lubricant in social situations. But there are risks that go with it, and some people may choose that they never want to drink. I used to, before I came to NIAAA, I used to go to the RSA reception, sometimes with friends who had a history of an alcohol use disorder, and get them a sparkling water and put a lime in it, and they had a good time at the party. And that was long before anybody served anything as an alternative for a party. So I'd like to see, stigma resolved, and that is part of should be an offshoot of everything that I've been talking about vis-a-vis the public. Alcohol use disorder and addiction in general is a brain disorder, but it's reversible. It's a reversible brain disorder. You can recover, but you can also be vulnerable to relapse. Just like you're vulnerable to relapse to shingles if you've had shingles, and you have to take steps to deal with that. Or diabetes or high blood pressure. You don't take your meds, you're going to relapse. And so, but we also need to get rid of the stigma of not drinking. So the stigma is working in these two different directions. And so, I hope to see a day when that occurs. I hope to see a day when if you need treatment, you get treatment. And the treatment is comprehensive, and it's not just 28-day rehabilitation, right? And it's tailored to the individual. And maybe there are some people who respond better to a camprosate. And, the Mayo Clinic chaps that I'm going to visit on Wednesday, who have identified those patients who do respond to a camprosate, that may end up being a blood test or something. And they say, you're a good candidate for a camprosate, or you're a good candidate for naltrexone, or maybe you're a good candidate for the next drug coming along. And there's lots in the pipeline, but we have to get them to the clinic. And then that's the other thing, that Ray Litton has been working on, our director of Division of Treatment and Recovery. We need better outcomes for clinical trials. And he's managed to get the FDA to accept, percent heavy drinking days in addition to abstinence. That's a huge breakthrough. But we want to make it even larger of a breakthrough by maybe using the WHO levels of drinking drop. Whether you drop one level or two level as an acceptable outcome. And that would facilitate the development of medications. Because, the pharmaceutical industry knows very well that if you're using abstinence as an outcome measure, you don't capture a lot of people, no matter how good the drug is. That's really kind of a pipe dream in some sense, that people are going to stay abstinent. And we know now that, many individuals need to be abstinent. But, as I said since day one, of my tenure at NIAAA. We look for abstinence, but we'll take anything we can get on the way, you know? So I think all of these are things that, you know, could be the future. And so we want people to be able to have treatment, and we need to, you know, we absolutely need to do something, and we're not yet, about adolescent treatment, about, you know, prisons, individuals who've been incarcerated, they get out, they start drinking, they overdose. I mean, it's an area that we need to put some energy. I didn't even mention that on Sunday, but you know, can't mention everything. But you know, there's still gaps in who, and then the diversity issue is huge. You know, and I did mention that on Sunday, but you know, I'm not even sure that the metrics we use to measure the harms of alcohol or the benefits of treatment with alcohol use disorder are culturally relevant. Do they really apply to different ethnic or culture groups? You know, this is a wide open area for young people, and I think they're starting to embrace it. And I think as we diversify our work group and our extramural, intramural communities, we're going to, you know, I met some people at the student luncheon yesterday, young people that are interested in working on these cultural specific areas of need, which are extensive. They follow the whole gamut. You know, I mean, there's horror stories out there where there was a paper published that showed that, you know, people who were diagnosed with the for an alcohol use disorder, certain ethnic groups would get a diagnosis for fewer criteria being met than white Anglo-Saxons, you know, which just reflects a bias in that, you know, and a stigma that's culturally specific that really needs to be addressed. So we have a lot of work to do in the domain of diversity. I'm just going to call it diversity, but it encompasses the whole gamut.
Yeah. For researchers at the start of their careers, do you have any advice to offer them?
Yeah, I mean, it's what I said yesterday at the student lunch. You know, there. So see if I can remember most of my points, but one is to find a mentor with a good academic lineage, and that doesn't mean it has to be Harvard or Yale. You know, it could be any school where the person has a good history of training individuals. And when I meant by academic lineage, I meant that, you know, there are people they've trained that have been successful. Another one I listed yesterday was, you know, to engage alternative mentors, which I call guardian angels. And these are individuals that can help you along the way. Joe Brady and Saul Snyder, both at Hopkins, were my guardian angels. They did enormous things for me, always took my phone calls, wrote letters for me. Joe Brady kept me out of the Vietnam War, along with my dad and Frank Holloway at Walter Reed in Division of Neuropsychiatry. So they got my file moved from a combat branch into a medical service corps. And, you know, I don't know whether I'd be here if it wasn't for those three gentlemen. And a third is be generous with publications and authorships and get them straightened out ahead of time before you start the study. It's the best to figure out who's going to be first author, who's going to be last author, or, you know, who's going to be share co-authorship. And you don't have to do it at the beginning, but that's the best way to do it. But, you know, be generous, because it's no skin off your nose if there are three authors as opposed to two authors. It really doesn't change anything, you know, in the end. And if somebody's contributed, then they should be honored. And then the last is, you know, connect with your peers, or I said network. And that really did seem to hit a nerve with everyone who followed after I spoke yesterday. So networking among your peers, because they're going to be people you grow up with and you're going to see at the meeting, and you see us old folks, you know, giving bear hugs to other old folks, and those are our buddies that we haven't seen in a long time. But RSA is an opportunity to reconnect with our old friends. So those are the four things for a happy life as a researcher.
Those are excellent things to live by. For members of the public who may watch this video, do you have anything that you'd like to communicate to them?
Well, that there is an entity called the National Institutes of Health. There are 27 different institutes. They all have websites, and they're all really, really good websites. And you can get an enormous amount of information from the NIH websites about health and how to have a healthy life. And so, you know, that would be one thing. And we at NIAAA have an excellent website as well, with lots of good information. And many of our resources are listed there, from rethinking drinking to college aim to you know, the treatment navigator to the healthcare, uh, the core healthcare professional resource. And so, you know, I urge the public to realize that they're paying for NIH through their tax dollars, but there's a resource there that could help any loved one who has any kind of disorder, but in particular, you know, for addictions, the National Institute on Drug Abuse, the National Institute on Alcohol Abuse and Alcoholism. Go there and you can find help and you can get things started. I think that's one critical thing to tell the public. Second is, addiction is a brain disorder and a body disorder. Alcohol use disorder is a brain and body disorder. It affects 200 different conditions, or causes or participates in 200 different conditions, from liver disease to the pancreatitis, uh, to fetal alcohol spectrum disorder, to gastrointestinal distress and gastrointestinal bleeds, even potentially, you know, dementia associated with aging. So you can go down this enormous list. And it's a window. Alcohol misuse is a window on so many other disorders. And so, you know, again, information is key to understanding your relationship with alcohol and the relationship of your loved ones with alcohol. And you know, it becomes as important as any other, maybe more important in some cases, than any other element of your daily life. You know, I mean, alcohol is pervasive, it's everywhere, it's part of our society. You know, we tried prohibition, that obviously doesn't work. So, you know, we have to learn how to negotiate as individuals with this substance. And some people don't want to drink at all. Some people shouldn't drink at all. If you're pregnant or thinking about getting pregnant, you shouldn't be drinking. You know, you should never be drinking and work with heavy machinery or climb a ladder or anything of the sort. I mean, there's so many things to learn about. If you're older and taking a herd of medications, you really need to be very careful about what medications you're taking with alcohol, because, you know, any sedative hypnotic, or like even an opiate, you know, 2 + 2 = 5, and you can end up with some severe respiratory depression, even overdose, and not know it. So, you know, and the last thing is that now, in collaboration with NCI, the National Cancer Institute has basically shown that, you know, 6% of cancers are caused by alcohol, and 4% of cancer deaths are caused by alcohol. And that usually gets the public's attention when I do public-facing lectures. So I will leave on that negative note.